Good morning, ladies and gentlemen. My name is Ameena, and with me, my partner Dana, are going to talk about our project role of the gut microbiota in autism spectrum disorders and inflammatory bowel diseases. Our PI is Dr. Ghazlan Bendris. 1 in 83 children in Qatar have been diagnosed with an autism spectrum disorder, also known as ASD, and it has become Qatar's national health priority. In addition to some genetic factors, environmental factors and lifestyle are thought to play a more important role in the pathogenesis of this condition than previously thought. There is strong evidence to suggest that there is a bidirectional communication between the gut and the brain. And this mostly depends on the bacterial fermentation of dietary fibers and production of metabolites and hormones by the gut. And this can be disrupted in a dysbiotic state. Our study is the first pilot study conducted in the country, with the main objective being to analyze the feasibility of implementing a fusion clinical trial involving dietary intervention on autistic children in Qatar. If we want to run such a clinical intervention, we need to be able to answer two categories of questions. First, how is the awareness of the population in regards to the importance of the diet in managing autistic symptoms? And is the population ready to engage in clinical trials that modulate the gut microbiome? Secondly, we need to understand the microbial profiles. We need to optimize our methods of recruitment, stool sample collection, extraction, and sequencing, and have preliminary data which could confirm the microbial diversity shifts that were observed in other populations. For this, we used a combination of online questionnaires and seminars to address the awareness and readiness among health professionals and the general public. Writing literature reviews was also an important way for us to raise awareness among scientists. For the other leg of the study, we collected stool samples from 55 individuals, which were divided into 3 cohorts. One was the autism spectrum disorder and the attention deficit hyperactivity disorder group. The other group was the IBD and the IBS, which is inflammatory bowel disease and irritable bowel syndrome group. And the third was the healthy controls. We used Fisher brand stool collection kit, which was easy to use for the participants to provide us with their stool. We then extracted microbial DNA using the QI agent kits. And then these samples were sent to CD Genomics for next generation 16 sRNA and ITS sequencing, and phylogenetic analysis was performed. We then used this data to compute descriptive statistics and correlational analysis using R and SPSS. And these are the key findings from the surveys. If we look at the proportion of individuals with obsessive compulsive and anxiety disorders among each group, we observe that there's a higher proportion among the ASD, ADHD, and the IBD, IBS group. This, again, supports the gut-brain interaction that we were hypothesizing. Second, if we were interested to analyze the diet type of the population, and among the 112 participants, most of them in the 3 groups have a diet which is rich in processed grain-based products. We also wanted to know about probiotics, as it is a way to correct dysbiosis. And we wanted to know if the participants were taking probiotics. And as you can see, more than half of the individuals that could potentially benefit from these are not actually taking them. 30%, 50% of the ASD, ADHD, and the IBD, IBS groups compared to the controls. In terms of awareness, we also wanted to know about the antibiotic exposure among the groups. And what we found, which was really important, is that 58% of the ASD, ADHD group and 38% of the IBD, IBS group had several courses of antibiotics during the first 3 years of life. This is something that should be monitored in future studies. In terms of awareness, among 79% of health professionals and 54% of the public were aware of the importance of gut microbes in pathogenesis of disease. And this is something we should increase. Finally, we cannot run a clinical trial if there is no willingness to engage in them. Here we can see that 90% of the participants reported to be ready to engage in dietary intervention, and 70% even to engage in fecal transplant, which is another way to modulate the gut microbiome. Interestingly, we noticed that health professionals seem to underestimate the willingness of the public to engage in such trials. Can you hear me? Hello? Can you hear me? Can someone tell me?
Yes. Yeah.
Okay.
Go ahead.
So now we'll be moving on to the microbial profiling results. And we can see that our data confirms what was shown in other populations, which is the presence of specific biomarkers in the gut,
with 29 operational taxonomic units for IBD and IBS and 25 for ASD and ADHD. Now, the originality of our study also comes in because we studied the microbiome of this, which is often neglected in other studies. And we can see that this was very fruitful because 1,638 taxonomic units were identified for ASD and ADHD, and 775 were identified for IBD and IBS. Now, the main key and expected finding for the study is the loss of biodiversity in the gut among the disease groups. And we can see the lowest among the ASD and the ADHD group. And now Qatar, for the first time, is confirming the same trends that have been shown in other populations. The decrease in biodiversity translates into the absence of millions of bacterial and fungal species in the autistic individuals. So we created a database that's consistent of 7 bacterial species and 106 fungal species that are all significantly less abundant in the autistic group in comparison to the controls. And we can see some of them displayed on the side, one of which is the Bifidobacterium longing, a very important bacterium in the gut, which is found 3.4 times less among the autistic group, further signifying that biodiversity in the gut is correlating with a healthy state. Samples of the ASD and the ADHD group did not only show decreased abundance, but they also had overgrowth of some bacterial and fungal species. We have samples of 2 right on the side from the order Clostridialis for bacteria and the Codnea order for fungi. Now, one of the key findings of this study is a fungi from the Willemia order, which was found 37.2 folds more abundant in autistic individuals in comparison to controls. Now, this species is unidentified and has not been reported previously in literature, highlighting the significance of this research and the significance of this finding for further investigation. Due to these findings, the loss of biodiversity has become an important therapeutic target to treat autism, having launched many clinical trials across the world that aims at modulating the gut microbiome for the treatment of autism. There are 2 methods to do such a thing. One would be fecal transplant, and the second would be through dietary interventions and lifestyle changes. One clinical trial that published its results recently shows this results, where after 8 weeks of fecal transplant, we can say that the severe symptoms were reduced by half, and after 2 years, 40% of the autistic individuals had minimal to no symptoms. Now, of course, fecal transplant does not come without consequences and side effects, and hence we are proposing the use of dietary interventions as a less invasive, more conservative method. However, it will take a longer period of time for it to modulate the gut microbiome. So to wrap up, ladies and gentlemen, we can say that we have collected a set of very important data that allows us to say 2 important things. One, the awareness and readiness of the population is favorable for the proposition of such a clinical intervention. And we did this by using 3 reviews, 1 that was published, 2 pending publication, a seminar, a grand round, and 2 poster presentations. We also proved to you, ladies and gentlemen, that there is indeed a need to reevaluate the diet that autistic individuals have, specifically focusing on the processed grain-based products by exchanging them into fibrous foods and also introducing probiotics into the diets of these individuals. The study has some limitations, including a small sample size of 55. However, we'd like to reassure you that everything was well documented and supported. Similar to any other studies, the microbial profiles are very difficult to generalize because of the difference they can create on the composition in terms of population, techniques, and diet, all of which can affect the composition. Looking into the metabolome is also something that needs to be considered for future studies instead of just taxonomic data. Finally, the project is not only ending its step here, but have yielded 2 different projects looking into the probiotic formula for ASD and other methods to release anxiety, all that surfaced because of the results of this study. Finally, we are now in possession of an important set of data and a network of collaborators from CIDRA, HMC, the Child Development Centers, and Weill Cornell University that enables us to propose a clinical intervention on autistic children that aims at modulating the gut microbiome by involving a complete lifestyle change. And this would be the first clinical trial available for autism in Qatar and in the Middle East region. making Qatar a pioneer for such research in the Middle East. We would like to acknowledge QNRF for the fund, as well as Ibrahim Leswi and the pre-medical department at Cornell. Thank you very much, and we're open for your questions.
Thank you, Ameena, and thank you, Dana, for your very informative and clear presentation. I may ask now Dr. Mohammed to raise his first question.
Yeah, thank you, Ameena and Dina, for this nice presentation. I have a couple of questions and some notes to raise for you. I know this is a very complex type of research, and it has been going for all over the world, actually. Many people interested in this and the implication of microbial biodiversity in so many health disorders, including metabolic disorders and ASD and IBD, as you mentioned. Now, I have one of the limitations you mentioned, or you forgot to mention in your study, is that the cohort, the homogeneity of the cohort in your study is a very important issue here. What is the age of your subjects? Are they taking medications or not? Usually you look at the demographic area and type of food and diet they take. All of these are factors that influence the microbiota in evaluation and also in the intervention in the future, if you think to go ahead and design an intervention therapy. So just to check, one of the questions, can you explain for me from physiological and biological point of view how the dysbiosis in ASD patient may alter the gut permeability, and so the behavioral changes in those ASD patients and IBD patients?
Yes, so you are absolutely right. There are many factors that affect the gut microbiome. And that's why when we did our questionnaire...
Excuse me, we can't hear your voice.
Yeah, what Dr. Gerard mentioned is 100% true. There are many factors that affect the gut microbiome. And so when we did our questionnaires, we had an extensive list of questionnaires: the diet, the ethnic and nationality, their diet type, probiotic use, antibiotic. We have almost 26 variables that we took into account in our questionnaires, and it's a very extensive analysis. We are still going to do the correlational analysis on it, and these results are still pending because we're still working on them. But we tried to take as much variables as possible into account because we know that everything affects gut microbiome, and we wanted to make our associations more accurate. As for the effect on the gut permeability, so the microbiota composition of the gut, how they affect the human body is through the secretion of metabolites like butyrate, propionate, acetate, and all of these alter the gut's permeability by affecting the tight junction expression and the secretion of mucus. For instance, butyrate, when there's excess of butyrate when there's an appropriate amount of butyrate, it increases the tight junction expression and increases mucus production. This again decreases the gut permeability, so less of the shorty chain fatty acids and the metabolites are able to escape outside the gut and induce negative impacts like triggering immune reactions and neuropeptides like serotonin. Serotonin is found to be very important in the pathogenesis of autism. 90% of serotonin is actually produced from the gut. And it's very easy to understand like how this could potentially affect the brain. And that is why we took this as our project, because we know that there's such a strong connection between the gut and the brain and every other organ, to be completely honest.
That's very good. Plus the pro-inflammatory cytokines, remember these in the IPDs, they play important role. The last question, quick question, the interesting in your project is that you're finding about this Bacterial mycota fungi, which is very high in the ASD patients. Did you look into what role of this fungi could do in this metabolites productions and how that could influence the behavior and the health of those patients?
So we are the first ones to have identified this particular species as being that much increased in our population. So we haven't been able to find papers or research on it, but we want to work on it if we have a larger cohort, and if we can do, our future plan is to focus on a metabolic analysis instead of a species, a taxonomic analysis, so we can understand how exactly they play a role in the pathogenesis of disease.
Okay, thank you.
Thank you. I just have one question. You know that you're talking about the relationship between the autism and the microbiota. So what happened in the absence of microbiota during early life? So what will happen if there is an absence of microbiota?
So basically what our study has found is that the first 3 life is the stages where the brain and the gut are actually developing. So the brain and the gut are among the organs that even after we are born, they are still continuously developing. And the first 3 years of life is where they start maturing and developing even further. And studies have shown that with the decrease of microbiota among these stages and early exposure of antibiotics, there's further correlation with having autistic symptoms, ADHD, and finding other disturbances in the neurological aspects of these individuals. In fact, one of the bacteria that we have mentioned, the Bifidobacterium longum, when it was found in decreased levels, when it was found in higher levels, these individuals were seen to have less childhood disorders in comparison to individuals who did not have Bifidobacterium longum in their bacteria. So by exposure of antibiotics and decreasing the biodiversity among these individuals, you are predisposing individuals to having neurological symptoms such as autism or even conduct disorders in these individuals.
Thank you, Dana. The next, I think, Dr. Hisham Saber. Dr. Hisham.
Yes, thank you, Doctora. Thanks, ladies, for the very interesting presentation. I'm not of the field, so you'll have to excuse my ignorance of it. Your hypothesis is that there is a causal link between the microbiota, gut microbiota, and ASD. But the underlying suggestion or hypothesis is that it is the cause of ASD. Is there a prospect that it being the effect rather than the cause, that it is caused by ASD rather than the other way around? I know the literature that you showed improvement of the situation by altering or amending the microbiota, which supports your initial hypothesis. But is there a prospect or a chance of it being an effect rather than a cause of ASD? Thank you.
So we do agree that there is genetic basis to the disease. There is some, but as you can see, there's
your voice is very far. We can't hear you.
So I'll take the question. So yes, this could be a possibility, and this is mainly because when we're looking into the studies for autism and as well as ADHD, one of the most important comorbidities that children with autism have is GI disturbances. And when you have GI disturbances, so these, for example, children did not like, for example, to eat certain dairy products and other things which are considered probiotics for your gut. So it could potentially be a cause. Right now, we are establishing that there is, in fact, a correlation between dysbiosis and an autistic state. We cannot say that it is a cause. It is merely correlation at this point. However, a lot of studies have been showing that GI disturbances being a comorbidity in ADHD and ASD children could be an indication that there is a pathogenic link in it, or it could be, as you said, the fact that because they have this GI disturbances, they are not eating probiotics, causing this loss of biodiversity and reversal and hence. And there's also a study that was published recently by Nature that showed that autistic symptoms can actually be reversed. So the study was conducted on mice, and they found that after giving these mice probiotics, they found that there was reversal of these autistic symptoms. Also, a documentary that was recently we've recently watched on autism also showed that they interviewed mothers, and they showed that when the mothers started introducing probiotics into their children's diet, there was a massive improvement in their autistic symptoms.
Thank you very much. You answered that amply.
Thank you. I think we run out of time. Thank you, Ameena, and thank you, Dana, for a very interesting presentation.